Hello and welcome back. Today we're going to go over what is called facilitated diffusion and osmosis. Alright, so in the last video we looked at passive transport. This is another form of passive transport. So there are two types of passive transport. There's diffusion and there is facilitated diffusion. Neither of these two types take any energy to work. They work on what's called a concentration gradient, which means moving from high concentration to low concentration. That's what a concentration gradient means. Okay, so last video, the diffusion that we looked at means that molecules were able just to go right past these phospholipids. They could just move through them. They were small, they didn't have a charge, so they could just sneak on through those phospholipids. Well, water can't do that. It is too big of a molecule, and it is a polar molecule, meaning that it has positive and negative sides to it. So it's not going to be sneaking through these phospholipids at all. So they're going to have to go through a specific type of protein. This little red thing here is called an aquaporin. It has a little channel or a little hole in the center that allows water to move through. And it can go both ways. It can go from outside the cell to inside the cell, or from inside the cell to outside the cell through the aquaporin. Okay, this type of facilitated, the passive transport, and it's called facilitated diffusion. We're going to use an aquaporin, which is a molecule and that helps it go from directly through the membrane. It provides a channel for the particles to move in and out of the cell. Again, this does not take energy. It is a form of diffusion. Aquaporins are the name of these proteins, and osmosis is an example of this. Alright, so here is an example of osmosis, a diagram for you. Alright, so if I get this to click. Okay, there we go. Okay, again, aquaporin has a channel. Water, these are your red and blue molecules. Your cell membrane, made of phospholipids. And these big green things are sugar. Alright, so we have, let's say this is the inside of the cell, and this is the outside of the cell. So you look up here first and you notice that there are one molecule of sugar on one side, a whole lot, 3 molecules on the other. There is more water on the left and less water on the right. So your arrows are telling you that there are more water molecules moving inside the cell than there are going outside the cell. So they are diffusing through these aquaporins. Alright, so osmosis is the movement of water across the membrane. The water will go from high concentration to low concentration of water. Okay, that's important. You got to remember that. And again, this does not take energy. It is a form of diffusion. Alright, so first off, we have some new vocabulary terms. Hypertonic and hypotonic. The problem is, I have students that get confused because hypertonic refers to the solutes, in this case sugar. Alright, so we're saying hyper has more sugar molecules on one side than it does on the other side. So look at this first U tube. It is hypertonic on the right, there are more sugar molecules. And hypotonic on the left, there are less sugar molecules. Okay, so that is where those terms come into play. So we just said that water will move from high concentration of water to low concentration of water. So this first beginning tube, we have high concentration of water. There is more water than there are molecules. On the other side, there are more sugar molecules than water. So water is going to flow from high water concentration to low water concentration. Even though they look the same up here, levels, that does not tell you what concentration there is. You have to look inside the tube. Alright, so this is what actually happens. Water flows across this little membrane into the other side, and it's trying to equal out the number of water molecules. Once they get equal, the term becomes isotonic. Okay, we'll get to that in just a minute. So real quick, I'm going to show you, if this works, a demonstration of this happening. Haha, it works. Alright, so this is a setup, a lab setup. This is a type of tubing that allows water to pass through. The brown stuff in the center is sugar, like corn syrup, sugar. Outside is water. So looking at this, the inside is hypertonic, the outside is hypotonic. So the water is going to move from high water concentration into low water concentration. As it moves in, this column is going to move up. It started at 800 milliliters. We're already over 900 milliliters. And it's not even been hardly 10 minutes. This is as you can tell, the video's been sped up. Okay, so let's go about halfway and see what happens. Please work. Okay, there we go. Look, it's moved clear up to here. And then at the end, you have all this water moving into the tube and pushing the sugar up. You can also notice, if you paid attention, the color of the sugar, of the maple syrup, I'm sure it's maple syrup, is lighter because water has come into it. Okay, that's demonstrating osmosis, water moving across the membrane from high concentration to low concentration. Let me get back to my slideshow. Here we go. Okay, so we've looked at this. Let's look at these words. Isotonic, meaning that water is equal on both sides of the cell membrane. This is a blood cell, so water is going inside and outside equally in the blood cell. Same thing in a plant cell. Hypertonic means that there is more solute on the outside and the water leaves. Whoops, I didn't mean to click that yet. So the water moves out. That makes the cell look really shrunken down and odd looking. And this happens with plants. When too much water leaves the plant cell, the plant will wilt. Hypotonic means that there is less solute on the outside, and the water is going to move in to the cell. And eventually, if too much water moves in, the cell will explode. It will kill the cell. Okay? And same thing with the water plant. Okay, so this is the effects of osmosis on cells, on living things. Alright, I really hope they helped explain the second type of diffusion called facilitated diffusion. Osmosis, again, is an example of this. All right. You have a review sheet, so hopefully you've listened. I will also put this video up on your Google Classroom with the slideshow so you can look through and if you need to on your own and listen to this again. Hopefully this helps you out a little bit. Have a good day, guys.